It is the late 1800s, somewhere in a European hospital, and a patient is bleeding out on an operating table. Doctors know one thing that might save this person: blood from another human being pushed directly into their veins. They try it. Within minutes, the patient begins to seize, their kidneys start failing, and they die in agonizing convulsions. This was not a rare tragedy. It happened again and again, in hospital after hospital, for decades, and nobody understood why. Sometimes the transfusion worked perfectly. Sometimes it killed the patient almost instantly. There was no pattern doctors could see, no warning sign, no way to know in advance whether the blood about to be pushed into someone's arm would heal them or kill them. Physicians called it a coin flip with a human life. And for over a century, nobody could explain why blood, something that looked identical under every microscope available at the time, could behave so violently different from person to person. That mystery would not be solved until 1901, and when it finally was solved, it opened a door into something far stranger and far older than anyone in that operating room could have imagined. Because buried inside human blood was not just a medical classification system. It was a molecular record of plagues, migrations, and ancient survival stretching back over 60,000 years, written into our bodies before written language, before agriculture, before the first cities, before almost anything we call civilization. Something is written inside your blood that no passport can forge, no government can issue, and no religion can claim. It existed before your name, before your country, before the civilization you were born into. And for most of human history, nobody understood what it meant. Then scientists decided to do something that had never been done at full scale before. They compared the blood types of populations across continents, across centuries, and across the ancient bones of humans who died tens of thousands of years ago. What they found did not just stun researchers. It rewrote the story of what we actually are underneath the surface of our skin. There is a question that medicine has quietly sat with for over a century, a question so enormous and so uncomfortable in its implications that most scientists have preferred to study its edges rather than confront it directly. Why does the blood running through a farmer in rural Ecuador look genetically different from the blood running through a nomad in Central Asia, a fisherman in coastal Japan, or a herder in the Ethiopian highlands? And why does that difference seem to trace ancient paths that human feet walked tens of thousands of years ago? Why does blood remember things that people themselves have forgotten? This is not a question about race. It is not a question about superiority or hierarchy. Read that carefully, because everything that follows will be misunderstood if that premise is not firmly in place. This is a question about survival, about plague, about geography, about the brutal and invisible war between human immune systems and the diseases that tried to eliminate them. The answer encoded in our blood types is not a social answer. It is an evolutionary one, and it is one of the most staggering stories science has slowly uncovered over more than a century of research. Let's start at the true beginning, not the human beginning, the blood beginning. The blood group system was discovered in 1901 by an Austrian scientist named Karl Landsteiner, who noticed that mixing certain samples of blood together caused the red cells to clump violently while other combinations mixed peacefully. He identified 4 groups: A, B, AB, and O. This discovery, which eventually earned him the Nobel Prize, was immediately treated as a life-saving medical breakthrough. It told surgeons which blood could safely be transfused into which patient, finally explaining why some transfusions healed and others killed. For decades, that was considered the entire story. Blood type was a medical classification, a logistical safety tool, nothing more. But here is what most people never learn. That classification isn't a random biological quirk, like the shape of your earlobes or the color of your hair, a neutral variation nature happened to stumble upon. The distribution of blood types across human populations is staggeringly unequal, and that inequality maps with remarkable precision onto the ancient roads that disease and humanity walked together throughout history. The most common blood type worldwide is O positive, with a global prevalence of roughly 38 to 39%. But step back from that global average and look at national and regional data, and the picture becomes far more dramatic. Ecuador leads the world in O positive prevalence, with around 75% of its population carrying this type. In Armenia, by contrast, roughly 46% of the population is A positive, an entirely different genetic landscape. In Thailand, B positive reaches nearly 37%, the highest concentration found anywhere in Southeast Asia. These are not minor statistical footnotes. These are massive population-level divergences, and they did not happen by accident. Think about what that actually means. The same species, Homo sapiens, anatomically identical, cognitively equivalent, spread across the globe and arrived in wildly different places carrying wildly different blood. The immune architecture of an Ecuadorian farmer, a Thai monk, and an Armenian villager are measurably biologically distinct from one another. And those distinctions trace back not to how these populations live today, but to where their ancestors stood which diseases their ancestors fought and which individuals survived long enough to have children. But here is where it gets deeper. New genetic variants determining blood types emerged as Homo sapiens migrated out of Africa between roughly 70,000 and 45,000 years ago, driven by encounters with unfamiliar pathogens and unfamiliar environments. Read that again slowly. Blood types are not simply inherited markers passed down neutrally through generations. They are biological scars, the leftover evidence of ancient epidemics. Every time a new population of humans entered new territory, a new climate, a new ecosystem crawling with new microbes, the diseases native to that territory began quietly selecting for certain blood types over others. People whose blood left them vulnerable died at higher rates. People whose blood offered even slight protection survived and reproduced. Generation after generation, epidemic after epidemic, famine after famine, this invisible pressure sculpted the blood type map of every population currently living on Earth. They weren't examining fresh blood samples. They were reading genetic code pulled from ancient bones, teeth, and skull fragments, recovered from caves scattered across Germany, Siberia, and China. Within that ancient genetic material, researchers zeroed in on markers for 10 medically relevant blood groups, still used in hospitals today. What they found confirmed something extraordinary. Blood type diversity is not simply ancient. It accelerated sharply the moment humans left Africa and began encountering the wider world. Archaeological evidence suggests that during this exodus, groups of Homo sapiens paused and lingered on the Persian Plateau, a region wedged between the Zagros Mountains and Iran, for roughly 15,000 years. Researchers believe that stretch of time would have been enough for new genetic variations to emerge, variations underlying blood types found only outside Africa. Essentially, a genetic incubator period before these populations continued spreading deeper into Eurasia. 15,000 years isolated between two mountain ranges, while the wider world transformed elsewhere. This wasn't a simple rest stop along a migration route. It functioned almost like a laboratory, a crucible where blood was being quietly rewritten by disease and survival pressure, invisibly, relentlessly, across hundreds of generations. And then came something even more startling. Researchers traced a rare blood group known as the RHD genes variant 4, a form that originated in Neanderthals and made its way into modern human populations through interbreeding somewhere along the route towards Southeast Asia, more than 65,000 years ago. Let that land for a moment. There are people alive right now carrying a blood type variant that first appeared not in Homo sapiens, but in Neanderthals. The interbreeding between our ancestors and theirs didn't just leave traces in immune response, pain tolerance, and sleep patterns, as researchers have documented for years. It left traces directly in human blood. Separate research examining ancient Neanderthal and Denisovan genomes has confirmed that these extinct relatives already displayed the full range of ABO blood group variability seen in modern humans today, and that both groups carried blood group alleles still recurring in modern sub-Saharan African populations, further supporting their shared African origins. The blood moving through certain people's veins today carries the molecular memory of a species that went extinct roughly 40,000 years ago. This is not metaphor. This is measurable genetics, sitting quietly inside living human bodies. But bring this forward in time, because the implications are not merely historical. They are intensely, uncomfortably present. The relationship between blood type and disease susceptibility has been studied in fragments for decades, but a large-scale analysis out of the Karolinska Institute in Sweden involving roughly 5.1 million people began consolidating what smaller studies had been hinting at for years. Type A blood cells showed a 20% higher infection risk following SARS-CoV-2 exposure compared to type O during a pandemic that reshaped the entire world, killed millions, and crashed economies. The vulnerability was not evenly distributed, and part of that uneven distribution followed the ancient logic of blood type. The mechanism isn't mysterious. Higher ACE2 receptor levels in type A individuals appear to facilitate increased viral binding, particularly with certain variants. The virus doesn't choose its targets arbitrarily. It finds the cell surface architecture, offering the best entry point, and for people carrying type A blood, that architecture offers a slightly better foothold. Not a death sentence, but a statistical disadvantage that, multiplied across hundreds of millions of people, becomes glaringly visible in the data. A 2024 analysis of UK Biobank data found that people with blood type O were less likely to test positive for COVID during the pandemic's first wave and also carried a lower risk of cardiovascular complications following infection. This pattern extends far beyond COVID. Non-O blood types show elevated pancreatic cancer risk, type A by roughly 32%, type AB by roughly 51%, and type B by as much as 72% compared to type O. Cancer, not infection. The molecular architecture of your blood, determined before you were born, shaped by epidemics your ancestors survived thousands of years ago, is quietly influencing your statistical likelihood of developing one of the most feared diseases of the modern era. For most people, this fact exists somewhere buried in scientific literature, occasionally cited in a health article, but rarely delivered with the full weight of what it actually means. Individuals with type O blood lack A and B antigens on their red blood cells, making them less prone to thrombosis and consequently carrying a lower cardiovascular disease risk compared to types A or B. Meanwhile, people with type A blood tend to show higher blood pressure and cholesterol levels than those with type B, placing them at greater risk for rapid arterial blockage development. On the other end of the spectrum, individuals with type AB blood show an increased likelihood of developing cognitive disorders later in life. Every blood type arrives with its own set of biological trade-offs. Every blood type is simultaneously a shield and a vulnerability, depending entirely on which disease you happen to encounter. This is exactly where the ancient logic becomes clear, because nature never designed a perfect blood type. It designed survival under specific local conditions, and those conditions varied enormously across the planet. Consider type O. It's considered the most ancient and widespread blood type, reaching frequencies of 75 to nearly 100% among some indigenous populations of the Americas. In some of these populations, type O is essentially universal, carried by nearly every person. Why? Because the populations that crossed from Siberia into the Americas brought predominantly O-type genetics with them, then spent tens of thousands of years relatively isolated across two continents where local selection pressures reinforced O as the dominant type. Pre-contact Americas was, in a very real biological sense, a world shaped almost entirely around the immune logic of type O blood. Then European contact happened, and with it came smallpox, measles, and typhus, diseases European populations had built partial immunity to over centuries of prior exposure, and against which indigenous immune systems were devastatingly unprepared. Historians estimate the death toll of European colonization across the Americas fell somewhere between 50 and 90% of the indigenous population, tens of millions of lives. The debate over exactly how much of that catastrophe was driven by biological vulnerability rather than warfare, displacement, and famine remains genuinely unresolved. But the blood type data raises an uncomfortable question researchers have only recently begun asking carefully. Was the biological architecture of indigenous blood, evolved in isolation and optimized for the diseases of the Americas, simply never trained to fight the pathogens of Eurasia? And did that gap become a factor in the catastrophe that followed? This question extends far beyond the Americas. The global distribution of blood types is, in a very real sense, a map of which populations encountered which diseases and survived them, and that map carries consequences that are anything but purely historical. Look at sub-Saharan Africa, where the data reveals something that upends common assumptions. In malaria-endemic regions of the continent, where recent global malaria reports document hundreds of millions of cases and hundreds of thousands of deaths annually, overwhelmingly affecting children under 5, the blood type landscape has been shaped for thousands of years by one of the deadliest parasites in human history, Plasmodium falciparum, the organism responsible for the most lethal form of malaria. The research connecting blood type to malaria resistance is complex and occasionally contradictory, as any honest review of the science will admit, but the broad pattern points toward something real. Blood group O appears to offer some protection against severe falciparum malaria, largely because O-type red blood cells show lower rates of a dangerous clumping process called rosetting inside blood vessels, while types A, B, and AB appear to show increased susceptibility. Rosetting, where infected red blood cells clump together and block circulation, particularly in the brain, is a key driver of deadly cerebral malaria, and it appears to occur more efficiently in non-O blood types. The implication is staggering. In regions where malaria has been endemic for thousands of years, natural selection has been relentlessly, invisibly culling individuals whose blood type left them more vulnerable to the parasite, gradually increasing the frequency of type O blood across those populations over millennia because the people who survived to have children were disproportionately carrying it. Your blood type, if your ancestors came from malaria-endemic regions, may in part be a biological inheritance passed down from the survivors of ancient epidemics. This is the brutal logic of biological trade-offs. Every adaptation protecting you from one threat may expose you to another entirely. Nature never offers true immunity. It offers a negotiated balance, and that balance depends entirely on the environment your ancestors happen to be living in. This means the blood type distribution of any population is essentially a biological record of which diseases that population fought most often throughout its history. A population with high O frequency likely survived generations of malaria. A population with high A frequency likely survived something O blood was less equipped to handle. Both populations carry these molecular signatures forward forever, passed down into the veins of descendants who will never personally encounter malaria, who will never encounter medieval plague, who live in climate-controlled buildings eating food their ancestors could never have imagined, yet who still carry the immune decisions made by the dead. The rarest is AB negative, found in less than 0.5% of people globally. That rarity isn't a curiosity. It's a quiet crisis waiting to happen in the wrong hospital at the wrong moment, since AB negative's extreme scarcity creates real challenges for emergency transfusions and donor matching that modern medical infrastructure is only beginning to fully address. Zoom in from global averages to national data, and the patterns start to read almost like a narrative. In France, roughly 37% of people are O positive, and nearly 36% are A positive, while only around 8.5% are B positive. That low frequency of type B blood in Western Europe isn't accidental. It reflects the relative historical isolation of Western European populations from Central and South Asian populations, where type B blood is far more common, believed to have proliferated among nomadic populations of the Eurasian steppe, the same populations connected to the Mongol Empire and the great Turkic migrations that swept across interior Asia for centuries. Where those populations traveled and where they eventually settled is still traceable, in part, through the lingering frequency of type B blood found in those regions today. Population geneticists have long treated blood type mapping as one of the most underused tools for understanding human migration history, building on decades of earlier fieldwork, collecting blood type data from populations worldwide long before modern DNA sequencing existed, work that has since been confirmed and refined by ancient genomics, now armed with ancient DNA analysis, modern population genetics, and enormous data sets pulled from millions of living people. Scientists are assembling a picture of human blood type distribution that functions simultaneously as a medical resource and an extraordinary historical document. These findings meaningfully expand our understanding of human evolution while carrying direct applications for medicine today, from transfusion safety to pregnancy care. An insight into the ancient genetic origins of blood groups may eventually help researchers find entirely new approaches to fighting diseases connected to specific blood types. There is also a present tense urgency here that health systems are only beginning to reckon with. In the United States, roughly 53% of the Latino population carries O positive blood, compared to only around 37% of the Caucasian population, a 16 percentage point gap between two large demographic groups living in the same country. Meanwhile, roughly 8% of the Caucasian population carries O negative blood, of the universal donor type, compared to only around 1% of the Asian population. O negative is what saves lives in a mass casualty event, when a trauma center is flooded with patients of unknown blood type and there's no time to test and match. If the surrounding population skews heavily toward demographics with lower O negative frequency, the statistical odds of having enough compatible donor blood on hand drop sharply. This isn't hypothetical. Blood banks worldwide manage supply chains with direct reference to the demographic makeup of their donor pools, and the mismatch between who donates and who needs transfusions, driven in large part by blood type distribution across different populations, is a genuine logistical challenge with real life or death stakes. In the United States alone, someone requires a blood transfusion roughly every couple of seconds, and these transfusions collectively save millions of lives every year. The blood type distribution of your own region may quietly determine whether compatible blood is available the moment you need it, and that distribution traces directly back through history, migration, and the ancient survivors who carried specific molecular markers forward into the present. And there's another layer researchers are only now beginning to pull apart fully, because the ABO system was never the only blood group system that mattered. Scientists studying malaria, HIV, COVID, cancer, and cardiovascular disease are increasingly focused on systems beyond ABO entirely, including the Duffy system, the MNS system, the RH system, and the Kell and Diego systems. These blood group antigens are increasingly recognized as major modulators of disease susceptibility, shaping how parasites invade cells, how immune systems respond, and how severe an infection ultimately becomes. The picture was never as simple as 4 letters. It was always a far more intricate symphony of molecular markers, each one carrying its own ancient bargain struck with disease and survival. The Duffy antigen system is perhaps the most dramatic example of this. Individuals who are Duffy negative, a trait overwhelmingly common among sub-Saharan African populations, are almost completely resistant to infection by Plasmodium vivax, another malaria parasite, responsible for illness in hundreds of millions of people worldwide. The Duffy antigen, sitting on the surface of red blood cells, functions essentially like a door handle that vivax uses to enter the cell. Without that door handle, the parasite simply cannot get in. The near-universal absence of the Duffy antigen across many African populations stands as one of the clearest examples of natural selection acting directly on human blood in response to a single specific pathogen, a molecular victory carried silently, unannounced, by hundreds of millions of people today as an inheritance from ancestors who survived something genuinely terrible. But beneath all of this sits an unresolved and genuinely unsettling question. We are looking at blood type distributions scattered across the globe and seeing the fingerprints of ancient disease. We are seeing the patterns left behind by the plagues, parasites, and pandemics of the last 100,000 years. Future pandemics will almost certainly do the same. Future diseases, whether emerging naturally or otherwise, will interact with human blood type distributions in ways that are partially predictable, partially understood, and enormously consequential for public health planning. A population that was overwhelmingly O-positive 2 generations ago may, through immigration and intermarriage, now carry a significantly altered blood type profile, with real consequences for organ matching, emergency blood supply planning, and disease risk modeling that health systems worldwide are only beginning to track properly. And, underneath all of it, runs a question science still cannot fully answer. We understand blood types evolved in response to disease pressure. We understand they vary by population in patterns tracing ancient migrations and ancient epidemics. We understand they correlate with differential risk for some of the most devastating diseases of the modern era. This is not chaos, and it is not coincidence. It is the story of human survival, written in the one language that outlasts every other record humanity has ever kept. The language of biology, of immunity, of blood itself. And the most important chapter of that story may not be the one already written. Your blood type is their story, passed down without ceremony, without explanation, without your consent. The real question is not whether that story exists. It's whether you're finally ready to understand what it's been trying to tell you all along.